My name is Mónica Santos, and I'm a principal investigator at the Center for Neuroscience and Cell Biology at the University of Coimbra. In this project in particular, we aim to understand the role of the neurotrophin-3 TRAC-C pathway in the formation and extinction of fear memories. Fear is a basic emotion, a set of defense mechanisms that have evolved to allow us to avoid or deal with danger. So, fear is good. However, maladaptive fear underlies many psychiatric disorders. Patients show impairments in the formation and in the extinction of fear memories that often manifest as an exaggerated response to fearful stimuli or exhibit a fear response towards neutral stimuli. In this scenario, understanding how fear memories are formed and extinguished is of uttermost importance to the discovery of both pharmacological and non-pharmacological approaches towards treatment of this group of patients.
In this project, we developed a model to study fear extinction that is based on the classical Pavlovian fear conditioning paradigm. The NMDA receptor subunit composition is linked with different synaptic plasticity events in the amygdala. In the amygdala of extinction success mice, higher levels of gluN2B containing NMDA receptors are associated with weak long-term potentiation and robust long-term depression, while in the amygdala of extinction failure mice, higher levels of gluN2A containing NMDA receptors are associated with strong long-term potentiation and no long-term depression. We observed that extinction success mice show higher TRAC-C activation than extinction failure mice in the amygdala. Importantly, exogenous administration of neurotrophin-3 to the amygdala of extinction failure mice was sufficient to rescue fear extinction, and extinction failure mice treated with neurotrophin-3 showed a reduction in fear levels similar to those observed in extinction success mice. Finally, we proved that neurotrophin-3 TRAC-C exerted their function on fear extinction through the gluN2B containing NMDA receptors, resulting in a weaker long-term potentiation and the accompanying fear extinction.
Results obtained with this project led us to propose a working hypothesis. The activation of amygdalar TRAC-C with extinction training leads to the accumulation of gluN2B containing NMDA receptors in amygdala synapses, which in turn impacts amygdalar synaptic plasticity by weakening long-term potentiation and triggering a robust long-term depression, leading to the successful formation of a fear extinction memory. When extinction training does not trigger amygdalar TRAC-C activation and the subsequent cascade of events, mice continue to show high levels of fear. Our results identify a role of neurotrophin-3 TRAC-C pathway in setting inter-individual differences in fear extinction, and puts forward this pathway as a potential target for the development of treatments for fear-related disorders. If I had to define the BIAL Foundation in a word, it will be empowering.